So again, this is a virus. This is called a bacteriophage, and like I said, a phage is something that kills something. So this is a bacteriophage, so it kills other bacteria. It actually, and we use these actually in research. If we want a bacteria to do our bidding, our evil doings, like make insulin, we can actually put the gene, the DNA of insulin, into this virus, and then that virus will then go into the bacteria, and the bacteria start producing insulin, which is pretty cool if you're diabetic. So, like I said, truly, it's actually a 3D model here. So, it's actually an icosahedron, which is a fun geometric term for 20-sided little object. Okay. Yeah. So, again, but we don't really care about like all those little sides because we want to look on the inside. So, really, what we have here. So, really, what we have here. The outside is what's called a protein coat. Okay, so go and label it. It's called a protein coat. So a virus is made up of basically three main parts, primarily only two, but sometimes three. The first part is called the protein coat. And it's just like a coat that covers your body. Okay? When you're cold, it's the same thing. The virus, now remember, viruses are not cells. So what covers the outside of a cell?
The cell wall.
Either the cell wall, and inside the cell wall would be the cell membrane. So for an animal cell, we don't have a cell wall, we don't have a cell membrane. So the protein coat is that outside covering. Now there's nothing else on the inside except for either double-stranded DNA, single-stranded DNA, or a double or single-stranded RNA. Okay, so go ahead and draw it in. Yep. So that, right there, is some kind of genetic material. I'm going to get really fancy here. We're going to call that nucleic acid. Okay? So on the inside of this protein coat is this nucleic acid, because what does nucleic acid tell us? Or what does it do? What does DNA do for our body?
Makes us who we are.
Yeah, it makes us who we are. So that DNA in there is going to say, okay, in this virus, this determines what that virus is. And based on the shape of the DNA, if it's double-stranded or single-stranded, if it's RNA or DNA, if it's single-stranded or double-stranded RNA, whichever one it is, determines what it affects. Okay? So this is a bacteriophage. They're most of the time... Good. So yeah, so again, here's a plant cell, right? And so all these structures are all within a regular animal cell as well as plant cell. But yeah, so the cell wall, the chloroplast, and then the large vacuole all makes up that cell. Okay. And again, remember, this is a parasitic relationship because the virus will actually go and attach to the cell, and then it will infect it. Okay, it'll actually be the parasite on it.
The Golgi apparatus, it helps carry stuff through the cell wall.
It shows one method of attack. I attach to the cell, serve vision like a cat. Check my genes inside, use my thinking machinery to make my genes and proteins. Reproduce authority. Step one, tail, five strand, the membrane on the wall. Check my genes, the cell is gonna fall. Three, use the victim, enumerate some ribosomes, synthesize the proteins, replicate my genes. The short tail, piece, and capsule cell, the cell, the stop them all together without putting all my projects. This is science.
Yeah? It's good, right? You can buy that on iTunes, by the way. It is good, right? It's actually on Classroom right now, so if you can watch the rest of it. Oh, we'll watch some more later.
I feel like at some point Eminem's gonna come out and start.
That is Eminem. That's Eminem in like...
In 2030.
Yeah, that's Eminem in the next like 20 years. It looks just like that. He's going to start singing science rap videos. It's going to be awesome.
Oh man.
So, Anyways, here's what I want you to do.
You know how you do that? You know how you do that? Shower and cold.
So, but here's the basic idea. Okay, so we have HIV, and HIV. The whole goal of HIV is essentially to shut down your immune system. If it shuts down your immune system, you will get sick. And so people don't die from HIV; they only die from other infections like pneumonia and the common cold, from flu, from being cut, those kind of things. Okay, so you've got this. Here's step number one. Step number one: the virus binds to a receptor protein on the host cell. So the virus binds to a receptor protein on the host cell. Now, we've talked about, and it talks about this whole like lock and key idea. Where else have we seen this idea of a lock, a specific key fits in a lock? On a door, yeah, on enzymes. You remember how an enzyme had a specific key to fit into a specific lock? That substrate fit into the active site in the enzyme, and only that specific substrate worked on that specific enzyme. Like lactase only works on lactose sugar. Maltase only works on carbohydrates. And so, and if we change the shape of that protein, if we heat it too much or we add too much pH, it's going to make that lock all kinds of messed up. So it's the same thing here. So there's step one. Step two.
Other cell surface proteins called co-receptors, not shown here, allows the virus to fuse with the cell. The lipid membrane of the virus incorporates into the cell's membrane, while the viral core enters the host cell.
All right, so here you go. So here's step number two. Step number two, the viral core. The viral core, and we can comma genetic material and protein coat. So the viral core, comma, genetic material and protein coat, comma, enters the host cell. Okay, so the viral core, which includes the genetic material and the protein coat, enters the host cell.
The viral RNA into DNA. The viral enzyme reverse transcriptase copies the RNA into complementary DNA or cDNA.
All right, so here's step five. Step five. There's step four. Sorry, step four. Step four is reverse, and I'll spell it up here in the front. So reverse transcriptase is used to turn the viral RNA into viral DNA. So reverse transcriptase is used to turn viral RNA into viral DNA. And again, remember, since it says ACE, it's a what? It's an enzyme. OS is sugar, ACE is enzyme. Yeah, turn viral RNA into viral DNA. Because remember, the cell doesn't have, the code is not in RNA, the code is in DNA in the nucleus.
Reverse transcriptase has a high error rate and frequently leaves mutations in the copied DNA. The mutations result in variant forms of HIV, which allow HIV to evolve quickly. The viral RNA is destroyed by another viral enzyme called ribonuclease H. Reverse transcriptase then synthesizes a second DNA strand, using the first strand as a template. The double-stranded viral DNA enters the nucleus with several types of viral proteins, including an enzyme called integrase. Integrase enzymes splice the viral DNA into the host cell's chromosomal
So you got step 5? Okay, so step 5. The viral DNA merges or enters, viral DNA enters the nucleus and merges with the host DNA. And this animation uses the term commandeer. Before it can commandeer the cell, so it's almost like a pirate. Right? The pirate boards your ship, takes out the captain, and now it becomes part of the, now that the pirate is now the captain of the ship, it's going to make the ship do what it wants it to do. That's what the virus is doing. It's a parasite on the host cell. So the viral DNA enters the nucleus and merges with the host DNA.
So if the virus gets past the membrane, is there any
Step 6, the host DNA codes for new viral RNA. The host DNA codes for new viral RNA. Codes, or makes, to make new viral proteins. Codes, C-O-D-E-S. Oh, the host DNA codes for new viral DNA. To make new viral proteins. Again, step 7. The new viral RNA and the viral proteins assemble near the cell membrane. And then step 8, near the cell membrane. And then step 8, the virus emerges from the host cell. Yep, emerges from the host cell.